Taxonomy of bacteria is very, very challenging because there are so many types of bacteria in this world. So in the beginning, it was messy. It's messy because there was no system. Any microbiologist that finds a bacteria and isolate that they identified differently, or they think it has a different criteria, gave a different results, biochemical results, then they name it as a different bacteria, or worse, sometimes they give it their own name and say it's a new bacteria. Okay, so it was very messy, but now they have made it very systematic. Okay, for any bacteria to be new, or any bacterial species to be accepted as a new species, it has to be published in certain journals. Not all journals, but certain journals only. And then you have to have that bacteria placed in one of the uh, microbiological deposit uh, centers. The one that you probably are familiar is your ATCC, American Type Culture Collection. Okay, so you must deposit it there. There are others. Uh, there's one in Germany, one in Japan. These are the ones I'm familiar with. I think if you publish in these journals, then you will get your bacterial species accepted. The journals are the International Journal of Systematic and Evolutionary Microbiology. Okay, uh, IJSEM, International Journal of Systematic and Evolutionary Microbiology. So this journal publishes new bacteria, uh, reclassification of a new genus. But in order to publish in these journals, you have to do the full set of biochemical tests against related bacteria and against certain standards. You have to do the 16S, full 16S sequence. You have to do the melting curve. Uh, you have to do a SEM and a TEM picture for micrograph. So there are lots of steps you have to do before you can submit it to the journal, and only after the journal accepts your paper, then the journal will include it into as a new bacteria. So other journals are Journal of Bacteriology, Journal of Applied Microbiology. Yeah. So there are some journals that are accepted for taxonomy purposes. Okay, not all journals are acceptable. So PLOS one, no. Some journals, no. Okay, don't do not think that if you publish in let's just say uh, Malaysian Journal of Science, or you publish in Science Malaysiana, a new species, then you think it will be accepted as a new species. No, it has to be through the proper channels. Okay, this is the only way that the microbiologists can control the taxonomy of this bacteria. If not, it will continue to be very messy. Okay, so in taxonomy, you have your taxonomic rank from the highest to the lowest, from domain, kingdom, phylum, class, order, family, and genus, and to species. So from domain, the most inclusive and the highest rank, to the lowest rank. Okay. So in bacteriology, when we talk about bacteria, the domain will be bacteria, kingdom bacteria, then the phylum. These are some of the phylums that we have. These are the phylums that are considered gram negative. Okay, Protobacteria, spirochetes, cyanobacteria, chloroflexi, chlorobium, mycoplasmas. Although mycoplasmas officially do not have a cell wall, but because it doesn't have any cell wall, when you do gram staining, it will stain red. So that's why they place it on. I included it in this slide. Good, fusobacteria and bacteroidetes. So these three are more likely to be found in anaerobic microbiology. So anaerobic microbiology is a microbiology field that focuses on anaerobes. Okay, because it is very difficult to handle anaerobic bacteria, it has become its own field in itself. Okay, so because you need a lot of you need specialized equipments, you need specialized procedures to handle anaerobic microbiology. So even in our program, uh, we I do not think there is anyone doing active research on anaerobic microbiology because yeah, it is quite difficult to. Okay, so the largest phylum is actually the Protobacteria. So under Protobacteria, you will have your alpha, beta, gamma, delta, epsilon Protobacteria. Okay, different classes. And and under order, you'll have different for the initial part of this course, we will be covering the Gamma-Proteobacteria. Under the phylum Protobacteria, we will cover the class Gamma-Proteobacteria. Why gamma? Gamma-Proteobacteria is where most of the bacteria that you can, whenever you culture something on a plate, there is a high probability that it is Gamma-Proteobacteria because Gamma-Proteobacteria are the easiest to culture on artificial media. Okay, so from the Gamma-Proteobacteria, you have different orders. So, and then from different orders, you have your family. So the difference between this, you can detect from the naming itself. For order, it ends with a LES, less. Okay. For family, it ends with a CEAE, CA. So the first group of bacteria we'll be studying is under the order Enterobacteriales. Under the family, Enterobacteriaceae. Okay, and the genus is Esterija Coli. So, so when you look at this name, Enterobacteriaceae, so you know this they are talking about family here, and these are the different bacteria species. So under Enterobacteriaceae, the main criteria that you can use to separate them is whether they are lactose fermenter or non-lactose fermenter. So if you're familiar with your McConkey agar utilizes lactose sugar. So those that grow and form pinkish reddish colonies, they are lactose fermenters. Those that are colorless are non-lactose fermenters. Okay, so we will cover E. coli first, Esterija coli. So it's the most frequent human pathogen. Okay, it is frequently found in clinical settings in clinical labs. So a lot of the procedures, a lot of the SOPs when isolating for bacteria in clinical samples will always include a McConkey agar, a blood agar, okay, will always include these two agars. Okay, so from there, immediately you will be able to differentiate between lactose fermenter and non-lactose fermenters. So E. coli is the main lactose fermenter here. These are also lactose fermenters. Salmonella and Shigella are lactose negative, okay, but very important also. Then these are under a different, uh, they are under a different family. They are actually under Yersinia, okay, but under the same order, these are under the Enterobacteriales order, okay. So these are all under different orders. So these are a new group of bacteria that is known to cause uh, heart disease, infective endocarditis, inflammation of the heart due to bacterial infection. Then we also have a group of bacteria, the escape. They are the Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, Enterobacter species. So these are the bacteria that are highly resistant. Okay, they are commonly found to carry antimicrobial resistant genes, and they are very important in hospital infections or nosocomial infections or hospital infections. So they cause a lot of problems in hospitals. Okay. So E. coli, Esterija coli is under the family Enterobacteriaceae, genus Esterija. Okay, so even under species, you can have variety or types, and you can have strains. Strains are usually a lab-based naming system. Okay, the way you name a bacteria in your lab. Okay, so that's why you can have different strains of the same species in a lab. So this is so Enterobacteriaceae is under the order Enterobacteriales. Again, it's an enteric bacteria. Enteric bacteria meaning it lives in the intestinal tracts of animals. Okay, it's mostly facultative anaerobic gram-negative rods. It's non-spore forming, catalase positive, oxidase negative. Okay, so oxidase negative is a very important identification step. This is an identification test to differentiate Enterobacteriaceae from others. For example, Vibrio-Naceae, which is positive, Pseudomonadaceae, which is also positive. So oxidase is an important step for identification. Then it ferments glucose in the OF test, the oxidative fermentative test. Okay, it grows well on artificial media. So it is a important contributor to intestinal infections. There are over 41 genera with hundreds of species in the family Enterobacteriaceae. Okay, then the most important characteristic is the lactose breakdown, whether it ferments lactose or not.